One of the things we learn when we get started in the research process is how much we don't know, or how many things we need to carefully consider in order to make an informed decision. Competent cells can be a great example, especially when starting out. Choosing the right competent cells for different applications is a decision that does require a lot of consideration. So to help you, we have compiled a list of some criteria for consideration when selecting competent cells so that you know some of the important factors up front that you need to evaluate potential cells for. Really quick though, I want to mention that these considerations focus on E. coli competent cells. Some of them are transferable to other types of cells like Agrobacterium, but when using different types of cells, there may be other considerations to keep in mind. Some things to help determine which route to take is the equipment, transformation efficiency needs, budget, and the needs of your cells. That needed transformation efficiency can definitely be a big factor depending on what you're doing. Electrocompetent cells provide better transformation efficiency, so if high transformation efficiency is a primary need for your experiment, then you probably want to lean in that direction rather than choosing the heat shock transformation method. We actually have a really quick video that goes into more detail about the pros and cons of both transformation methods, so rather than repeat a lot of it, maybe just take a look at the other video after this, and we'll have it linked below for you, of course. One thing I do want to say though is that whether you're using the heat shock method or electroporation, transformation requires meticulous, careful work. Aside from transformation method, another consideration when choosing your cells is genotype and genetic markers. When researching competent cells, you'll often find a section in the product information with the genotype of that cell. And there's a whole nomenclature associated with this in order to understand the properties of that cell. I'm not going to get into that understanding genotype nomenclature in this video because it would need its own video. But we do have a resource, and of course we will have that linked as well. What you do need to know about genotype is that it contains information about the strain in terms of its important genes, mutation types, allele numbers, antibiotic resistance, insertions, fusions, inversions, and deletions. For instance, when it comes to mutations, the E. coli recA1 mutation in cloning strains are important because it prevents your clone DNA from recombining with the cell's genome. Even without knowing everything about genotype nomenclature, you can see in these GB5 alpha cells that the genotype has a recA reference. The whole point though is that genotype shows you certain properties, and given the needs of your experiment, you'll want to look at the genotype to make sure your cell is equipped to do what you need it to do. The next consideration you're probably familiar with is downstream applications. What are you doing further down the experiment, and how will your cell choice impact that? When it comes to competent cells, two downstream applications that usually follow are cloning or protein expression, and your cell choice will depend on which of those two routes you're going to take. We have a really cool decision map illustration that you can use to help determine what cells are more appropriate. So let's say your next step after transformation was cloning, then you're going to need to use strains specific for cloning. Some examples of cloning strains include GoldBio's GB5 alpha cells and GB10B cells. Both have important properties that make them appropriate for cloning, and we have a video on this that goes into more information about those properties. If you're cloning and working though with larger plasmids between 10 kb and 350 kb, our GB10B Pro would be more suitable. We also go into more detail about this in our article about choosing competent cells, which we'll have that link below for you too. We have lots of links. Let's say you want competent cells for genomic and cDNA library construction, then GoldBio's GB10B or 10B Pro would be good options with higher transformation efficiency and allows you to clone methylated DNA. Anyway, maybe your next step after transformation is phage display or protein expression. So let's look at these different scenarios and what cells would be ideal. For protein expression, BL21 and BL21DE3 cells are good expression strains because they lack certain proteases that degrade synthesized proteins. Finally, for phage display, GoldBio's TG1 phage display cells are ideal. In TG1 phage display cells, an AMBER suppressor suppresses the AMBER nonsense mutation. This is a mutation that inserts stop codons, which leads to truncated proteins. In the end, this is a good list of considerations to get you started with competent cells. It all really comes down, though, to what your needs are, what your expectations are, and what resources you already have available. We have a lot of information about competent cells on our website, as well as a really great selection of competent cells. Just visit goldbio.com to learn more, and I will see you in the next video.